Hello students, I am VJ. Let's start the new lecture of Biology class 10th chapter Life Processes. In the previous video, we have discussed about the different photosynthetic events, differentiation between the land plants and the arid zone plants, or that means the terrestrial plants and the desert plants on the basis of photosynthesis. Now we are going to start activity 5.1, and that is very important for board exam, so you have to focus a lot. So let's start with this activity. Activity 5.1. So any activity, first we have to mention the aim, for what purpose we are having this activity, कि किस बेसिक ऑब्जेक्टिव के साथ हम इस एक्टिविटी को पढ़ रहे हैं, क्यों पढ़ना चाह रहे हैं हम इस एक्टिविटी को. So let's focus on that. So aim of this activity 5.1 is chlorophyll is necessary for photosynthesis. Chlorophyll is necessary for photosynthesis. That is the aim of this activity. So for this, second is material required. What materials we have to take in that? First, we have to take a potted plant, with variegated leaf. Variegated means, variegated का मतलब होता है रंग बिरंगी. रंग बिरंगी का यहां से मतलब ये है that has more than one shade of green. That means light green, dark green, yellow. So all the shades of different shades of green we are taking in that, and the example for that, common examples are money plant and croton. These are the two plants, those are given in this activity, same activity as you see here, the croton and the money plant. Next, after taking this plant, what else we have to take, और क्या हमें लेना है? हमें लेना है methylated spirit, that is also the form of alcohol. Then we have to take the iodine solution. आयोडीन सॉल्यूशन के अलावा हमें लेना है ट्रेस पेपर, pencil. So all these are the basic requirements. So we have discussed the two important points about this activity. First is the aim and second is the material required. Next we need, next is the procedure. The very first step of each and every activity related to photosynthesis is de-starching. So step 1 is de-starching. De-starching, D means elimination and starch means already stored starch. So that means elimination of already stored starch. बेसिक पर्पस इसका ये है कि हमें कोई कंफ्यूजन नहीं हो कि एक्सपेरिमेंट के अंदर पहले से मौजूद स्टार्च बना लिया हुआ है, या जो अब हमने इस एक्सपेरिमेंट को किया है तब ये स्टार्च बना है। तो हमारे दिमाग में कोई एक कंफ्यूजन नहीं हो कि हमने पहले वाला स्टार्च के हिसाब से रीडिंग ले ली है ये, या नए स्टार्च की रीडिंग ली है। तो देयर शुड बी नो कंफ्यूजन, सो दैट्स व्हाई we first eliminate all the starch that is stored in the plant, and whatever the starch is produced, that is the new during the experiment, not the already stored starch. So first step in each and every activity related to photosynthesis is de-starching. Step second. Now, after this step, we put the plant in sunlight for 6 hours. अब एक चीज और ध्यान रखेंगे हम, डिस्टार्चिंग के लिए वी हैव टू पुट द प्लांट, कीप द प्लांट इन डार्क रूम फॉर 48 to 72 आवर्स। ये आपको बात ध्यान रखनी है, 48 to 72 आवर्स और 2 to 3 डेज, 2 से 3 दिन के लिए बिल्कुल अंधेरे कमरे में रखना है, ताकि सारा पहले से स्टोर्ड हुआ हुआ स्टार्च यूज हो जाए, यूज्ड अप। और जो भी अब इस एक्टिविटी में स्टार्च बन रहा है उसके बेसिस पर हम इस एक्सपेरिमेंट को कर रहे हैं। तो सेकंड स्टेप हमने किया कि पुट द प्लांट इन द सनलाइट फॉर 6 आवर्स। देन वी टेक द थर्ड स्टेप, स्टेप 3। अब स्टेप 3 में हमने क्या किया, वी प्लक अ लीफ, pluck a leaf and trace it with the help of trace paper. We trace the leaf with the help of a trace paper to mark green and non-green area. To mark green and non-green area. अब ऐसा करने के बाद now we move to the third step, and third step is step 3. Now we put the leaf in a test tube having boiling water. अब हमने इस लीफ को इंसर्ट कर दिया बॉइलिंग वाटर के अंदर। अब इसका रीजन है टू किल द सेल्स, टू किल द सेल्स, टू अप्रोच that there is no production of chlorophyll after we take it out from the sunlight. Next is the step 4. Step 4 is to put the leaf in methylated spirit or alcohol. But we have to avoid direct heating of this alcohol. as alcohol is highly inflammable and volatile. Inflammable means catches fire at very low temperature, and volatile means convert into vapors at room temperature. So for these two reasons, we have to boil methylated spirit or alcohol in water bath. That is the indirect heating. So in this indirect heating that is named as water bath, what should we do? We take a beaker and we heat it with the help of burner, and we take in this beaker the water, that is water, and we take a test tube, and in this test tube we put the methylated spirit, that is methylated spirit, ठीक है, and then we put the plugged leaf. अब हम उस लीफ को इसके अंदर डाल देते हैं. So that is the plugged leaf, and we insert it in this boiling alcohol for few seconds, take it out, and by this, what is the purpose for that? Its purpose to remove chlorophyll. To remove chlorophyll. Now, alcohol solution becomes green and leaf becomes colorless. Leaf become colorless. All are the questions, potential questions which can be asked in the exam. Same questions आप देखेंगे आपके एक्टिविटी में भी दिए हुए हैं. देखिए, डिप द लीफ इन द बॉइलिंग वाटर फॉर फ्यू मिनट्स. आफ्टर दिस इमर्स इट इन ए बीकर containing alcohol. Carefully place the above beaker in a water bath and heat till the alcohol begins to boil. What happens to the color of the leaf? तो अभी मैंने आपको बताया leaf become colorless. What is the color of the solution? Solution become green. अब next क्या है? Next is what we do, take out the leaf and rinse off with iodine solution. अब क्या करेंगे next? Take out the leaf from alcohol solution and rinse मतलब होता है धोना, rinse with iodine solution. We wash this with iodine solution. अब iodine solution का purpose है, its purpose to detect the presence or absence of starch. Absence of starch. Presence or absence of starch. If there is blue-black color, so that means the presence of starch. If there is brown color, hence there is absence of starch. Up when the leaf has turned up or appeared on the some side, we see the patches of blue and black color, and other side we see the patches of, किसी हिस्से पे हम देख रहे हैं ब्लू ब्लैक कलर आया है और किसी हिस्से पर ब्राउन कलर आया. तो कैसे हम कंफर्म करेंगे? तो हमने ट्रेस किया था, वी कंपेयर द रिजल्ट विद ट्रेसिंग लीफ डायग्राम। जो हमने लीफ का ट्रेस वाला डायग्राम बनाया था, उससे हम इसकी कंपैरिजन करेंगे, मैचिंग करेंगे। तो जब आप मैचिंग करेंगे, यू विल फाइंड दैट ब्राउन कलर विल appear where the non-green areas and blue black color appear where we have the green areas. So that will confirm only the green areas contain chlorophyll, not the non-green areas. So similar question is asking or is there in the activity, that what can you conclude about the presence of starch in various areas? So here you write in green areas there is a presence of chlorophyll because here photosynthesis will take place and blue black color is appeared. But in case of the non-green areas there is no chlorophyll, hence brown color is appeared after putting iodine solution, and that confirms chlorophyll is necessary for photosynthesis. I think you have understood this activity very well. Let's move to the next section. We have discussed this activity along the questions, and that is a very important source for case study based questions. So you must go through this activity very carefully. Next is, you can see on the next page the this diagram of stomata. In stomata, these are tiny pores on the surface of epidermis. These are the epidermal derivatives. Another thing, these meant for stomata are meant for gaseous exchange. Stomata are meant for gaseous exchange. So for gaseous exchange, stomata are present. So if we take the open stomata diagram, so you can see here that these are the stomata, guard cells, and these are the outside subsidiary cells, and then we have the outside epidermal cell. So this is the diagram. So that is the epidermal cells. These are the, ये एपिडर्मल सेल्स हो गई, ये हो गई आपकी सब्सिडरी सेल्स, and this is the guard cells, and that is the stomatal aperture. So through this stomatal aperture, oxygen, water molecules come out and CO2 get in. So this is gaseous exchange, how plants take up the CO2 and release O2 and H2O. That is the major function of stomata, that is named as gaseous exchange. Next thing is opening and closing of stomata. Another very important question for board exam, opening and closing of stomata. So let's take opening. So opening of stomata and closing of stomata. So opening takes place during day and closing during evening or night. So what happens during day? Due to photosynthesis in guard cells, due to photosynthesis present in guard cells, as guard cells has chloroplast, the concentration of solute is increased and outside cells have more water. So water moves in from outside cell. तो पानी जो है बाहर की सेल से गार्ड सेल्स में आना शुरू हो जाता है. इवनिंग में देयर इज नो फोटोसिंथेसिस, hence outside water is less and solute is more due to transfer of food during evening. Hence water moves from guard cells to outside. तो यहां आप देख रहे हैं, इट इज रिटन, if you focus on this, so you will notice here that opening and closing of the pore is the function of guard cells. The guard cells swell का मतलब end osmosis. Water enters from outside to inside guard cells, flows into them, and that become turgid. गार्ड सेल्स टर्जिड हो जाती हैं, फूल जाती हैं पानी से लबालब, और कॉजिंग स्टोमेटल पोर टू ओपन. अब वहीं आप दूसरा सेंटेंस देखेंगे, सिमिलरली द पोर क्लोजेस इफ गार्ड सेल श्रिंक. तो श्रिंक का मतलब है यहां पर फ्लैसिड, और फ्लैसिड नेचर कब होगी? इफ देयर इज एक्स ऑस्मोसिस. सो पानी जब गार्ड सेल से बाहर की तरफ मूव करेगा, सो दैट इज एक्स ऑस्मोसिस. आप इस तरीके से समझिए, जब पानी अंदर आएगा तो इस तरीके से गार्ड सेल्स बाहर की तरफ फूलेंगी एंड देयर इज ओपनिंग ऑफ स्टोमेटा. जब पानी गार्ड सेल से बाहर की तरफ निकलेगा, देयर इज क्लोजिंग ऑफ स्टोमेटा. So that's a very important question for 1 or 2 mark in board exam. So far we have discussed activity 7.1, the related questions and the values points, and then we have discussed the opening and closing of stomata along with the function of guard cell. So that's all what I'm going to give you this in this lecture. Stay connected to all other different lectures related to this chapter, और इसी तरीके से हम बायोलॉजी को आसान करते रहेंगे और बायोलॉजी का डीजे वीजे के साथ बजाते रहेंगे. थैंक यू फॉर दिस लेक्चर.